Hello and welcome to BioRules video lessons. Today we are starting a new subtopic in biochemistry. We are going to be talking about nucleic acids. And if you live in this planet in the last 50 years or 60 years maybe, when you see an image like this, you are immediately going to say DNA. Everybody on this planet that's old enough to be able to say words, look at this and say, okay, that's a DNA. Okay? So, nucleic acids involve DNA and other molecules, and just for you to have an idea, where can we find DNA? In our cells, eukaryotes, DNA is going to be found coiled in chromosomes inside the nucleus, inside of the cell. Every single cell of your body, with a few exceptions, are going to have your entire DNA. So every single piece of it has all the information to make a new one of you. Okay, let's take a look on the structure of this molecule, because this is a very, very special molecule. It's amazing, beautiful, it's something fantastic. So, nucleic acids make many stuff. DNA is an information molecule. Actually, from the DNA we can make many other stuff. We can make proteins that can make other stuff. So basically all the life is coded into DNA that are going to be making many, many other stuff, okay? DNA is going to be whole information, and that's what we're going to be talking right now. This is a piece of DNA, and those little bars connecting these two coiled strings over there are those stuff like that thymine, this adenine. These here are bases. They are nitrogen bases, and the information is going to be into these chemicals. If you have one chemical like this thymine, you have one information. If you have another one like that adenine, you have another piece of information. So DNA is going to be holding or carrying or delivering information, and that information is in this base over here. So let's take a look on the structure of this DNA. As we see here, DNA is a double helix. Basically we have one helical stuff that goes like this, and then we have another one in the other side. Now they are kind of parallel, we are going to see that in a moment. That goes on the other side, and now they coil, make a double helix. DNA is made of two tapes. You have one side, another side, and they are kept together in a double helix. If we take a closer look there, we are going to see that DNA, you have two strands. Of course, if it's a double helix, you have two strands making DNA, and these strands are going to be like this stuff here. We have a closer look, we are going to see one of the strands of DNA. So this here is one of them. One strand of DNA that have these balls, these pentagons and those arrow stuff over there. If we take a closer look on there, we are going to see what is the base of the basic structure of the DNA, which is the nucleotide. In the same way that glucose is the basic unit of starch, cellulose, glycogen and others, in the same way that amino acids are the basic structure of proteins, the nucleotide is the basic structure of nucleic acids. Basically, nucleic acids are going to be repeating nucleotides one after the other after the other. And these nucleotides, if you take a closer look on that, it's going to be something like this. The ball is actually a phosphate group of phosphorus with oxygens. The pentagon is a sugar, which is a ribose or a deoxyribose. And that little arrow on the side is one of the nitrogenous bases that you saw before. In this example, we are seeing an adenine. Okay? So let's take a closer look on this sugar, because this sugar is going to be important even to determine if you have a DNA or an RNA. So the typical ribose that you saw in biomoles unit is this here. We have 5 carbons connected with an oxygen here in the middle, and they form this pentagon. Ribose has OHs all over the place. So you have an oxygen there, another one oxygen here, another oxygen there. This guy have an oxygen over here, and this guy has an oxygen over there. So riboses have an oxygen connected to every one of those carbons. Deoxyribose means a ribose with less oxygens or without oxygen, misses an oxygen in this guy over here. So in this carbon, you have a missing oxygen in the deoxyribose. Okay. Another thing that's important here is for you to know these numbers. We have carbon number 1, that's the one that's going to be connecting to the base, to the nitrogen base. You have carbon number 2, which is the one that will have oxygen in ribose and no oxygen in deoxyribose. Carbon number 3 is one that's going to be connected to another nucleotide in the bottom. Carbon number 4 is just there. Carbon number 5 is the one that connects to the phosphate that you connect to another nucleotide on the top. So carbon 1, 2, 3, 4, and 5. For us here is going to be very important the carbon number 1 that connects to the base. Very, 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 very important carbon number 3 and carbon number 5. So if you know those, then this is going to be helping you a lot. Okay. Here we can take a look on the structure of DNA, and we have over here on the bottom you can see a sugar. This is a deoxyribose. I know that because DNA contains deoxyribose. And we have here a phosphate deoxyribose backbone. What does that mean? Phosphate deoxyribose backbone means this: phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, and so on. And here you have a phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose. You have a backbone. Hanging from there, you're going to have bases like that adenine, this cytosine over here, this thymine over there, this guanine. So this here, they are hanging from the phosphate deoxyribose backbone. And as I told you before, this here is carbon number 3. So this here is going to be the 3 end of this strand. And this one here on the top, this is the 5 carbon. So this here is going to be the 5 end of this strand of DNA. Okay? So this here is a basic structure of nucleotides being put together. And now you know deoxyribose in DNA. Okay? So we have there the 3 end, the 5 end, and the other way around we have the 3 end on the opposite side and the 5 end. We are going to see this here. Closer look. This guy here is the 3 end, and because we have here carbon number 3, is this one over here. So that's the one over there. And on the other side, we have the carbon 5 here. You're going to have carbon 1, 2, 3, 4, and 5. So this here is carbon 5, meaning that this here is the 3 end, is the 5 end of this base. Okay? So let's take a look. A closer look here on the nucleotides. We have the sugar in DNA deoxyribose without an oxygen over here, so missing an oxygen in carbon 2, the phosphate, and the base. What are the bases? Bases are nitrogen molecules with nitrogens and carbons. You have a carbon here, carbon here, carbon here, another carbon there, another carbon here. So this here is going to be making one of 5 nitrogen bases. So they are the cytosine, also known as C. You have the thymine, also known as T. You have the guanine, also known as G. And you have the adenine, also known as A. DNA contains those guys. And in RNA, instead of a thymine, RNA contains uracil. So it's another nucleotide over there. And here we also have classification of these guys. First on a level, knowing the cytosine, thymine, guanine, adenine, or even A, G, C, and T, that's more than enough. Or in the U, of course, that will be in the RNA. If you are a higher level, you should know the difference between a purine and a pyrimidine. So there are ways for you to remember this. I remember that pyramids, if you think on a pyramid like that, no, they make something like this, and the pyramids, they contain one base, like this here. They contain one square there in the bottom. So the pyrimidines here, they are pyramids, and they also have, if they were a weird pyramid here, they would have one kind of square in the bottom. So pyrimidines is one, and purines are going to then be the one that have 2. Pyrimidines 1, purines 2 rings over there. Which one are purines, which one are pyrimidines? So there's another trick for you to try to remember. I remember a student once made this for us. She said, her name was Gabi, and she said that GA, Gabi, are pures. So Gabis are pures, so they are pure. So GNA purine. So the others, the C, the U, the T, they are the pyrimidines. Whatever going to be used to remember this, have fun. As soon as you remember, high level, which one are purines, which one are pyrimidines, which ones are one ring, which ones are two rings, I'm going to be happy that you are going to be knowing this and you're going to be able to use it. Okay? So here we have all that stuff together, the nucleotide put together making a single strand, two strands together make a double strand, and here they twist around and they make a double helix. Here we see the iconic image of double helix DNA. Okay? So here. So let's take a look at the DNA structure in a close look. Here we have an adenine, just to be illustrating to us how things get connected. You have a phosphate, a sugar, and a base, in this case adenine. And then when we connect to another nucleotide, we are going to be connecting the phosphate, which is in the 5 and the 5' carbon, to the 3' prime, the 3' of the sugar, the deoxyribose on the top. And this connection is going to be a covalent connection, covalent bond, which is a very strong bond. So the backbone of the DNA, phosphate, sugar, phosphate, sugar, phosphate, sugar, is connected by covalent bond, which is quite strong, quite difficult to separate. Okay. What about the other side? In the other side here, I'm putting an adenine, but actually it doesn't go here. This should be a thymine. Okay, and they go on the other sides. They connect to each other, but they connect going the other way around. You have the 3 side on the top, they are going to have the 5 side, the 5' on the bottom. So this is not adenine, but just illustrate here how they go side by side. One goes on one side, the other one goes on the other side. It will be a little bit more clear in the next slide, but just to mention here for you to see, these 2 strands are connected by hydrogen bonds, which are intermolecular bond, not intramolecular bond, and therefore they contain the 2 strands. Okay, it's a DNA molecule, yeah, but actually DNA molecule is made by 2 strands, which actually it's kind of 2 molecules. So hydrogen bond connected to 2 sides of the strands, and that makes the 2 sides, the 2 strands of the DNA to be quite easy to separate, but very difficult to break the backbone itself. So to break the strand of DNA itself is going to be very difficult, okay? To illustrate how things goes, we have here phosphate, sugar, phosphate, sugar, phosphate, sugar, with bases hanging there. And in the other side, quite parallel, but the opposite direction, which you call anti-parallel, you have the phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay? So this here is the way that they go. And we have here the phosphate and the sugar, making the basic backbone of the DNA, phosphate, sugar, phosphate, sugar, phosphate, sugar, and we have the bases hanging on in there. On the other side, we are going to have also phosphate, sugar, phosphate, sugar, phosphate, sugar. So they are anti-parallel, and we say that because if this here is the 3 carbon, is the bottom of the sugar, this here is going to be the 5 carbon. And in the other side, then it goes the other way around. This is the 3, and that one is the 5. So we say that this goes from 3 to 5, so this here will go from 5 to 3. They are anti-parallel, and there we have the bases connecting them, and the bases, as you can see here, they are going to have hydrogen bonds connecting them in here. In this slide, we can see again the anti-parallel 3' and 5' end. So in the other side, you have the 3' end in the other direction, and here the 5' end. So these guys, they are anti-parallel. In this stuff, you can see the adenine or the A connected to thymine, T, so this is A connects to T. And here we can see the guanine connects to the cytosine, so the G connects to the cytosine. And if you are paying attention, you probably realize that the A and the T, the adenine and thymine, they make 2 hydrogen bonds, 1 and 2 here, while the guanine and the cytosine over here, they make 3 hydrogen bonds, 1, 2, and 3. So this is illustrated here. This is something for you to know, 2 hydrogen bonds, and here we have 3 hydrogen bonds connecting them. Okay, so this will be quite important in the near future. Okay, so here you have diagram showing again. If this is a 3', can you guess the others? I hope that by this time you can already say that this is going to be 5, and this one will be 3, and this one will be 5 again, because they are anti-parallel. They go in opposite directions. Okay. Now we are going to be comparing the structure of DNA and RNA. DNA, deoxyribonucleic acids. So the sugar is a deoxyribose. RNA, ribonucleic acid. So the sugar is a ribose. This is one big difference between them. Just an oxygen per sugar, but it makes an entire difference. It makes the entire difference for the name, just for starts. The other thing that you can see here is that the DNA is a double helix. It's 2 strands, side by side, kind of helical coiling one over the other. And the RNA is going to be a single strand of nucleic acid. Another thing that you can see here is that both of them are going to contain the base adenine, which is green. Both of them contains the base cytosine, which is yellow. Both of them contain the base guanine connected to cytosine, so red connected to yellow. And the DNA will contain the base thymine in blue, which connects to the adenine. But the RNA don't contain this blue. You see that the RNA on there on the right contains only uracil. So this is a difference between DNA and RNA. DNA is ATCG, RNA is AUCNG. Okay, so let's take a look here again. We have thymine on the DNA, we have uracil on the RNA. This is a typical classic difference between them. And here, double strand, single strand RNA. Okay, so let's build a little table, which is the easier way for us to compare stuff. So comparing DNA and RNA. So in terms of nucleotides, which ones are there? If you said ATCNG for DNA, AUCNG for RNA, you got it right. Okay. Also, DNA contains deoxyribose, RNA contains ribose, and number of strands, DNA is double and the RNA is single. Okay. DNA is very, 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 very long, and the RNA is usually short. By short, I mean short comparing to the DNA. DNA can have millions of bases, and the RNA contains hundreds to maybe some thousands of bases, but so thousands is much smaller than millions. Okay. In terms of function, which is not comparing the structure of them, but if you compare the function of the DNA is to hold information. Those bases A, T, C, and G, they are going to be containing information. And the RNA, they can carry information, but they can also work as enzyme. They are used to copy DNA. That can be used for energy. ATP is a ribonucleic acid. ATP is RNA, is one nucleotide of RNA. So you have this comparison. Okay, so this is the basic structure of DNA and RNA. Very, very important to understand this, because we are going to be playing with DNA, building them, copying them, making information. So make sure, for your own sake, that you really understand DNA and RNA structure, all that we saw over here. If you're having problems, let's discuss in class to make sure that you nail this. Okay? I hope you had fun, and now take a look on the quiz, try to see if you really understood this. See you in class, bye-bye.